Okay. Actually, I made a question for this doing microbiology. The question was using a graph, describe the four phases of bacterial growth. Describe the four phases of bacterial growth using a graph. The question was carrying 20 marks. That was very, there are a lot of marks allocated on it. So, without wasting much of time, we have four phases in the growth of bacteria. We have four phases in the growth of bacteria. So we have what we call log phase, log phase, then stationary phase, as well as decline phase, or you can say phase of decline. So when you ask like that, say using a graph, describe the four phases of bacterial growth. Of course, you should draw a diagram like this one. Then when you're done drawing a diagram like this one, you should also have to be able to start now describing after just below the diagram. So the log phase, where you have to say the number of bacteria in the log phase, or you can say the number of cells, because a bacteria is just a component of cell. It's just a cell. It has got structures like cell membrane, the capsule, the flagella, the microvilli, and the other structures. So it's just a cell. So I'm saying in lag phase, the number of cells are not that enough. Meaning there are a small number of cells, but these cells or the bacteria in the lag phase, they are increasing in size. So the bacteria in lag phase, they are increasing in size. Why are they increasing in size? For readiness to undergo binary fusion. Binary fusion, because these bacteria, they multiply through binary fusion. So in lag phase, they will be able to increase in size so that they are ready to undergo binary fusion. And the number of cells in lag phase, or the number of bacteria in lag phase, they are not increasing. They are just small number of bacteria in lag phase. So that is the first phase. So there are just few things that you can mention. The number of cells are not increasing; they remain the same. Then the bacteria or cells are increasing in size, in readiness to undergo binary fusion, because bacteria, they multiply through binary fusion. That was lag phase, the first phase. Then the second phase, we have the log phase. Log phase, we can be able to see um, um, are reducing in size. Bacteria are reducing in size, or cells are reducing in size, but they are increasing in number exponentially. So an exponential word means rapidly, meaning in log phase, the bacteria are increasing in number rapidly as compared to any other phase, because they undergo multiplication at high rate. So that is log phase. The number of bacteria increases exponentially, meaning they increase rapidly, meaning cell division or bacterial division in this phase, it is of high rate. Then the third phase, we have the stationary phase. The word stationary, um, uh, it is coming from the word static, meaning things are not moving, they attain the equilibrium. So the cells, cell number remains the same. Cell number remains the same in the stationary phase. So here, bacteria are not multiplying, rather they are not even dying, but the number of cells remain just the same. You can even see from the graph how it is showing. It is able to show that it is in the straight line. That means that we are not increasing in number, we are also not decreasing in number. So stationary is the third phase of bacterial growth, where bacteria cannot multiply. They are also not dying, but the number of bacteria at that time remains just the same. And we have the last phase, which is decline phase, or phase of decline. So in this phase, you would see that the cells number are decreasing, meaning the bacteria are now dying. They're decreasing in number, isn't it? So that was uh, how you could answer that question, which was phrased like using the graph describe the uh, bacterial growth curve. So that's how you would answer. You have to first draw. When you draw, yeah, you have now to start explaining these phases one by one below the diagram. At that time, would have got it in correct